Opportunity to thank the organizers for inviting me and for organizing such a great meeting. As a non-immunologist, I've learned a great deal. What we are is a signal transduction lab, or if you want to think of it in a different way, as a quantitative cell biology lab. And what I'll talk about is related to some of the work that Rob presented this morning on lymphocyte migration. And what we're interested in is how signal transduction, so intracellular biochemistry, regulates processes like proliferation, survival, and migration, not just in immune cells, but in many different types of cells. And so we're working closely with the Duke BU group in their efforts to model the germinal center reaction by looking at lymphocyte motility. But really I want to talk more generally about cell motility and cell migration. Before I do that, though, I just want to emphasize that we have another part of our lab that works on signaling networks. So looking at how different pathways interact with one another. And it's a combination of biochemical measurements, live cell imaging measurements, and modeling of various types. Mostly biophysical style models. So why is it important to study cell migration? Well, I think everyone in the room can appreciate that cell motility is involved in a range of physiological processes as well as disease processes. So the example that I always like to give is in wound healing, because wound repair requires the coordination of many different cell types, and also coordination on different time scales. So very early on, you have sort of the inflammatory response, recruitment of neutrophils and macrophages, and these cells then secrete cytokines and growth factors that are directed towards other cells, most notably fibroblasts, which respond chemotactically to those growth factors and also proliferate very robustly. And by those processes, the fibroblasts invade the wound space from the adjacent dermal tissue over a period of several days. And this makes up what we refer to as the proliferative phase of wound repair. So it's important to understand the communication between leukocytes and mesenchymal cells like fibroblasts. And then later on, towards the end of the talk, I'll talk about some of our preliminary work with lymphocytes. And there again, you see in the germinal center reaction potential communication between lymphocytes and stromal cells. So again, these two different types of cells are important in just about every physiological process. So I'm going to make a distinction between two types of cell crawling behavior. On the left is a video of a neutrophil chasing down a bacterium. It's responding chemotactically, meaning it's responding to a gradient of peptides that are being shed by that bacterium. And in that way, it's able to follow the path of the migrating bacterium. This is the so-called amoeboid mechanism whereby leukocytes and certain other types of cells migrate very fast. They're unfettered by strong adhesion, so they're able to sort of squeeze through small spaces and move very rapidly. Contrasted with that is the so-called mesenchymal mode of motility, where you have much slower, adhesion limited, if you want to think of it that way, migration speeds. And one of the things you can see, I think, very clearly from these two videos was that in this case, the neutrophil was moving with a very well-defined polarity, with a well-defined front and a back, and that are maintained as the cell turns, whereas the fibroblast on the right has a number of competing protrusions and is not very strongly polarized. And that's going to be the main theme of this talk. Okay, I won't get into all of the details of all of the sub-processes involved in cell migration, but it is fascinating. I'd be happy to talk with anyone who's interested in more detail. But some of the highlights of what we're interested in are the spatial organization of the molecular componentry, some of the stochastic transitions that occur, particularly in relation between adhesion dynamics and cytoskeletal dynamics. And probably most interesting to us right now is the interplay between signaling biochemistry and mechanics of adhesion in the cytoskeleton. Now we're all familiar with cell tracking, as Rob talked about this morning, and some of the different models that you can abstract from cell tracks. So you can think about cell crawling as a persistent random walk that perhaps is biased by tactic gradients of various types. So chemotaxis would be migration that's biased by a gradient of a soluble factor, haptotaxis migration biased by an immobilized factor, and so on. So various external cues can bias this process. One thing that the cell tracking sort of throws away, however, is the shape changes that occur as the cell crawls. So the different structures that form that are associated with the protrusion of the cell, and also the the corresponding retraction at the cell rear. And I can just, in the interest of time, just point you to some of the work that's been done in morphodynamics of cell movement. Just showing you here a highlight from some previous papers in the Dictyostelium discoideum field. That's a social amoeba that is used by a number of investigators as a model system of chemotaxis, of amoeboid chemotaxis. Because you have genetic control and a lot of the advantages that yeast investigators thrive on. In terms of mesenchymal migration, there's also sort of this idea that the cell shape has something to do with how the cell crawls, so how persistently it crawls and so on. And other investigators have tried to link morphodynamics, particularly at the leading edge where the cell is protruding, with the intracellular signaling imaged using live cell microscopy. However, they're doing this usually on a very short time scale and a very limited spatial scale. So they're all about resolution. But the problem here in our mind is that the persistence time can be significantly longer than those time scales. So they're looking at things at the minute time scale, but when you want to look at slow-moving cells and how they turn, you really need to look at the hour time scale. So there's some technical challenges associated with that disparity. Okay, so what I'm going to talk about first is our work in fibroblasts, which is all published, so I can talk about it in detail. And the questions that I'm going to hope to address are as follows. How are spatiotemporal dynamics of signal transduction? And in particular, I'm going to focus on the phosphoinositide 3 kinase signaling pathway. It's not the only pathway that's important in cell motility. It's one of them, but it's what we've done the most with. And we're going to look and see how that's coordinated in relation to motility. And then later on, I'll tell you functionally what this pathway does in fibroblasts. And then the second question, which I'll get to later, is how is persistence maintained if motility is governed by short-lived processes? So signaling, cytoskeletal processes are all pretty much on the 1-minute time scale in fibroblasts. So how can the cell maintain a persistent directionality for 1 hour? The primary experimental technique that I'll be talking about, I'll be showing you, is total internal reflection fluorescence microscopy. And all you really need to know about it is that it selectively images the region of contact between the cell and the substratum. Okay? So it's as if we're looking at the footprint of the crawling cell. Now, for this reason, we can only look at cell migration on flat surfaces. So we can't use this technique, to look at, elegant in vivo experiments. So there is going to be some compromise here. And that's really an open question, probably over drinks or whatever. I'd be happy to talk about that. And the cells are going to be expressing a fusion protein biosensor that's comprised of a domain that binds to the lipid products of phosphoinositide 3 kinase. And that domain is then fused to GFP or one of its spectral variants. And so when there's more lipids produced in the membrane, more of our biosensor goes to the membrane where we're exciting the region of contact, and we see an increase in TERF intensity. So that's the pathway we're looking at and how we're measuring it. So we've done this in the context of both random migration and chemotaxis of fibroblasts towards PDGF. And one thing I think you can see from the random migration case is that PI3 kinase is localized very dynamically. So you sort of see these hot spots of signaling flare up in particular locations. You can probably guess that these locations tend to be regions where the cell is protruding. So that's perhaps living up to our expectation if this pathway is in fact important for cell migration. We see the same thing in chemotaxis. The source of the growth factor is up here. Initially, these cells being weakly polarized don't necessarily follow the gradient. Sometimes they just sit there. Sometimes they sort of eventually get smart, like this guy, and starts migrating with a very strong polarization aligned towards the gradient. Okay, and we did things like correlating a vector associated with the pattern of PI3 kinase signaling and correlating that signaling vector with the vector of cell movement. And what your eye just told you in the previous movie is that, yes, they do positively correlate very well. We can look at the autocorrelation of both the signaling and the movement vectors to get some information about the time scale associated with those processes. So we see that the autocorrelation holds with an exponential decay of something like 15 to 20 minutes. So these little hot spots tend to flare up and persist for about 10 to 20 minutes. So we're getting a little bit closer to looking at the directional persistence, but this was sort of just the first step, I would say. So to answer this question, really get at this question of the disparity in timescales, what Eric Welf did in my lab was to come up with a new way of analyzing the data. Well, not new, but new for us and different from what we had done in the past. And this was just recently published a couple of months back. So again, I'll try to go through it quickly. And what he did was to come up with a way of spatiotemporally mapping protrusion and retraction velocity and the pattern of signaling and also the shape of the cell. There is, of course, some compression of information thinking about the visualization of data that we heard about yesterday from Martin. But I think you'll agree that it was quite helpful in picking out what's going on. So specifically what we do is we determine the density of protruded or retracted pixels in a given time interval as a function of where they are with respect to the centroid. So we see a density of pixels as a function of the angle relative to a fixed vector, in this case the negative x-axis. And we can do the same thing with static images of the signaling, and then also pick out regions that are extended from the cell body as a measure of where the protruding structures are, and gives us some information about cell morphology. And this is just showing you the protrusion and retraction map. So now we're plotting as a function of position, and then time going from the top to the bottom, sort of in kind of like a kymograph style. And you can pick out things almost immediately. The red here blotches are the protrusion, blue is retraction. So you can see that even though there's a particular region where protrusion and retraction are occurring, that you have punctate spots on this map. So there are particular regions of the leading edge where protrusion is happening, interspersed with some retraction. So 2 steps forward, 1 step back sort of thing. The other thing you can see is an example like this, where you have a region that's predominantly protrusive and then switches. Something that an amoeboid cell would almost never do. It basically throws it in reverse and becomes a tail. But I think what really leapt out at us, especially in a cell like this that is doing this behavior a lot, is that the leading edge of the cell tends to branch, okay, and produce multiple branches. And if you compare that behavior with the localization of signaling, you see a very nice correspondence even by eye. And these tend to occur in these morphological extensions that we're picking out. And so there seem to be certain regions where protrusion and retraction seem to occur, privileged regions. And without getting too much into the details, we're fairly certain that it has to do with stress fibers being on the sides of the cell and preventing protrusion in those places. Okay, so what is PI3 kinase signaling doing in this process? Well, what we did was we used both a pharmacological inhibitor and also a dominant negative construct. And both pretty much do the same thing. They prevent branching. In the case of the inhibitor, it stops branching in its tracks. If you then quantify the number of initiated branches, PI3 kinase inhibition does not grossly affect the initiation of branches, rather the successful propagation of those branches. So you're still putting out new branch processes, but without PI3 kinase signaling, those new branches are unstable. Okay? One thing that was somewhat surprising, but perhaps not now that we had established this particular role of PI3 kinase, the stage at which it's important, is that PI3 kinase signaling is elevated after the onset of protrusion, not before. Okay? So you start to get protrusion, then you get the highest increases in PI3 kinase signaling, as indicated by this negative time shift in the correlation between the two. So this was averaged over many cells and obviously many time intervals. So it sort of makes sense that you need PI3 kinase after you've initiated a new protrusion. It then stabilizes it and allows it to have a chance of successfully propagating. And what do these branches do? Well, here's just one extra thing. You can actually induce protrusion semi-artificially by using a photoactivatable form of constitutively active RAC. Same RAC1 that Eberhard referred to in his lecture. And when you do that, you can focally activate that. You can recruit PI3 kinase signaling to that region. Okay, so again, protrusion first, then recruitment of PI3 kinase. And what does this do? Branching of cells mediates turning. Okay, and if it successfully propagates, and this is just showing how, in one instance, but we saw this in many cells and characterized this, but this is just a stereotypical example showing where we give a PDGF gradient to the cell, so a chemotactic gradient, and the cell, as it turns, it does so in a way that's biased so that it aligns properly with the gradient. Okay? And this is just showing the branch here on the spatiotemporal map. The green dots is the position of the steepest gradient, and ultimately the branch that wins ends up being aligned almost perfectly with the steepest gradient. Okay, so this is the model that we have for mesenchymal directional persistence. You start off with a stable morphology of a head and a tail, pretty much going in one direction. Then you initiate a branch, and that's unstable. Okay, without PI3 kinase signaling, that new nascent branch is very likely to retract. Okay, with PI3 kinase signaling, however, you are able to give some stability to that nascent branch and allow a chance for that branch state to propagate. Now, of course, the branch state is not inherently stable because it's self-limiting, for one thing. So as you keep propagating, you end up with basically being bipolar. And that too is unstable. The cell has to pick one way or the other. And which one it picks seems in our hands to be biased by external cues. And we have some ideas about what signaling processes may be going on there, most likely involving ARP2/3 mediated branching of the F-actin network. Okay. Now, how much time?
Total 10 minutes, so 5 minutes.
Okay, great. So now we're starting to adapt these approaches to analyze lymphocyte migration. And in working with these cells now for a short while, I can really appreciate how difficult these cells are to work with. We started with cell lines, and we've gradually moved on to human T cells from peripheral blood, and now mouse splenocytes, both T lymphocytes and B lymphocytes. And of course, we're interested in cell movements during the germinal center reaction, primarily looking at B cell migration as they migrate as naive B cells into the dark zone, and then ultimately following chemotactic cues into the light zone where they encounter follicular dendritic cells. And I probably know less about that than most people in the room, so that's all I'll say about it. But what we're doing is, here we're not looking at cell signaling because we're not able to express our favorite probes in these cells, at least not yet. So what we're doing is labeling the plasma membrane of the cells with the lipophilic dye, and fortunately we're able to see the contact area very well under turf, at least in a lot of the cells, and we can observe the changes in cell shape as the cells move. This is the work of Aster Liu in my group, and she loves the fact that these cells move very fast. So, 30 minutes, the experiment's done, move on to the next cell, whereas my other guys are waiting 8 hours just to look at one fibroblast. And this is the work I'll tell you about, again, very preliminary, is random migration of T cells, human T cells, on ICAM1 and CXCL12. Just randomly coded on the surface. And again, we're adapting our methods, so the same kind of spatiotemporal maps for protrusion and retraction velocity, again, red protrusion, blue retraction. And then also identifying regions, the morphological extensions, which could be the protruding pseudopods and also the retracting uropod. So pick those out. And just show you a movie real quick that Eric Welf, a postdoc in my group, very talented, he sort of picked out a way to visualize this. So the red little blobs here are the morphological extensions, and then this sort of ring here is showing you the protrusion and retraction velocity, and so you can see sort of dynamically how that evolves. And so we're still trying to figure this out, but I'll give you our sort of first thoughts on this kind of data. We've got we've imaged a pretty good number of these kinds of cells, as well as the mouse cells. And what you can see, if you delineate the regions of of protrusion and retraction, you can see that the front of the cell, there's a lot of activity, okay? And there, in particular, there's a lot of branching. So that's something we were ready for, based on both on our fibroblast work and the work in dictyostelium. But by comparison to this sort of chaotic environment at the front, the rear is quite smooth, okay? And you do see, though, interactions where protrusions sort of start out at the front and then move their way to the back. Those are these sort of diagonal lines that connect the front and the back. And you can sort of ask yourself, well, what happens when the cell makes major changes in direction? And, well, one of the characteristic behaviors is that a certain protrusion will stall at the side of the cell, and this then serves as a pivot point that ultimately merges with the cell tail. So again, the cell maintains polarity, so it doesn't just throw it in reverse. It has to move the tail, you know, sort of as it changes its overall directionality. Okay. And where we're going with this in terms of modeling is a statistical description of this with Tom and Grace, and then also a more mechanistic, mechanical description, and we're still trying to sort that out. So let me leave you sort of with what the main conclusion is that fibroblasts and lymphocytes are different. You know, it's biology, right? So in terms of, they both do this sort of branch and pivot turning, but they're very different. So in mesenchymal cells, you see frequent switching between protrusion and retraction. So it doesn't have a committed polarity at all, really. You see privileged regions of protrusion and retraction. And for those reasons, then what you see is a radial branching, okay? And in conjunction with the retraction of the rest of the cell, this would be where you would place the pivot point, if you will. And then ultimately you make up to a 90-degree turn, and then the cell picks either, you know, one way or the other. Actually, the pivot point would probably be a little bit, you know, the next centroid would be a little further up. Amoeboid cells, at least the lymphocytes we've looked at, but I think other indications from other labs looking at neutrophils and dicty are consistent with our findings, is that they have a strong polarization between the front and the rear, but they have a free range, sort of what I call free range protrusion at the front. So these protrusions can slide over laterally and explore the entire front of the cell. And they branch at a much higher frequency than they actually turn. Okay, and if we were to look and see, sort of then what happens then is that you slide over the leading edge and it rotates. Again, do this sort of lateral movement of this, or wave-like movement of the pseudopods. And then the tail, just mechanically, the tail is forced into, you know, sliding around in concert. And we think that myosin 2 is playing an important role mechanically in mediating that. Okay, so that's all. I tried to acknowledge the people who did the work along the way, and I'm happy to take questions.
So, any questions? So when you measure PI3 kinase, you mainly use pH domain biophysics construct, right?
Yes, we use the we use the pH domain of AKT.
Right. In that case, you know, that distribution of pH domain is also, you know, in part depends on PI3 kinase, but at the same time it depends on P10 distribution patterns as well. So you have like positive regulator and negative regulator as well. So have you considered that about P10 functions in your?
Right. So the when I say PI3 kinase signaling, really what I'm referring to is an accumulation of the lipid products, which of course depends both on the phosphorylation of PIP2 and is also affected by the dephosphorylation of the product. So if you were to take a lipid phosphatase such as P10 and either tune down its activity or tune it up, that also affects, and we're certainly sensitive to that. And we've published some papers that suggest that the lifetime of the lipid actually is different in different places of the cell. That's dating back now to 2005 that we did that analysis. So I think that is a very good point and could definitely affect the magnitude of the signaling. Now, if we do block PI3 kinase, though, everything comes down. So it is dependent on the enzyme.
I guess that competition might be even more important for this branching effect. Whenever cell has to give up one side, and maybe there must be some dominant negative effect on that side, whereas when cells decide one direction, there must be some dominant positive effect of the other enzyme.
Right. The fact that it has such a striking polarization is suggestive of some kind of feedback loop, right? And we have no reason to doubt that. There's good indications for mechanistically how that could happen, and there's many modeling papers out there. Mostly they're looking at different kinds of cells in formulating their models, but there are a lot of models on PI3K, P10 regulation, especially in DICD where the knockouts are so clean and the role of P10 is very well established.
Yes, we have to cut off.
Okay.
Anyway, thank you very much.